Hello friends, welcome back to my course on Microbial Biotechnology. Today we are going to start module number 10, which is about microbes in medical biotechnology. We are going to discuss in this lecture the production of biopharmaceuticals using this recombinant DNA technology, mostly focused on the production of enzymes, antibodies and other therapeutic proteins. So this lecture is divided into two broad sections. The first sections will be dealing with the basics of recombinant DNA technology for those who are new to this technology, and section number 2 will be for the application of this recombinant technology in the production of pharmaceuticals. So let's start with the basics of rDNA technology. In this figure you can see bacteriophage from Staphylococcus aureus, where phages are used as vectors in rDNA technology. What are these vectors? We will be discussing about them soon. So these particular section has the following content. Number 1, we will begin with a discussion about the advantages of using recombinant DNA technology, then what are the basic steps of this technology, and then isolation and purification of DNA, which is the most important first step in the recombinant DNA technology. Then cleavage of DNA into particular sequences. How do we cut down these large DNA molecules into smaller fragments and also vector molecules, because engineering is all about cutting and joining. Here we will be showing you how we cut DNA of interest and paste them into carriers called vectors, which are also basically DNA molecule. After cutting these fragments, we paste these into these vectors, is known as the ligation. So we will have a discussion about the ligation of these DNA fragments. Then once this vector is constructed, which is basically a carrier having a DNA or a gene of interest, we need to introduce these recombinant DNA product into suitable hosts. Hosts basically are living cells where these vectors will multiply, thereby also multiplying the recombinant DNA. For production of protein therapeutics, the gene that we clone into the vector needs to be expressed first, transcribed and then translated. So we will also have a discussion about the expression of recombinant DNA in the host. Then we will discuss about E. coli as an effective expression system and the various engineering strategies involving E. coli, and then identification of host cells that contain the recombinant DNA. So let's start with a very basic introduction of recombinant DNA technology or rDNA technology. This involves bringing together genetic material from diverse sources, resulting in sequences absent in the original genome, and is therefore sometimes called chimeric DNA technique. DNA sequences utilized in constructing recombinant DNA molecules can originate from any species. By employing recombinant DNA technology and synthetic DNA, virtually any DNA sequence can be generated and introduced into living organisms. Proteins resulting from the expression of recombinant DNA within living cells are termed recombinant proteins. So let's have a look into this general scheme for recombinant DNA technology. So here we have a bacterial cell which has extra chromosomal DNA called plasmid. So using these plasmid we construct the vectors, or some of these are already ready to use vectors. Now on the other hand, let us take an example of an eukaryotic cell which also has DNA, but this is the nuclear DNA and that we call as a chromosomal DNA. Now both these vector DNA and chromosomal DNA are subjected to restriction digestion with the help of restriction endonuclease. So here you can see that due to the digestion by restriction enzymes, this vector has opened up. We made a cut in the vector. Similarly, this DNA will be digested into small pieces, okay, and then by the restriction endonuclease activity. Now these small fragments of DNA we can insert into this vector by a process called ligation. So, here these are the product as a result of this entire procedure, and these particular vector molecule carrying the gene of interest is known as the recombinant DNA molecule. Now, these particular recombinant DNA molecule is sent inside a host cell, which is for example, bacterial cell, and by the process of transformation, and then we get the transformed bacterial cell. Now, these vectors having the loaded DNA inside may actually simply keep on multiplying because the plasmid vectors are autonomously replicating units, and while the vector multiplies, the DNA which is cloned into it will also multiply. So, thereby we will be able to increase the copy number of these gene of interest by this process. If we add certain DNA sequences which helps in the expression of the gene, for example, if it is a protein coding gene, we can also further have the protein product resulting out of this process. According to the interests or according to the needs, we may use simply cloning vectors, which helps us in increase in copy numbers, or we may also use expression vectors, which helps us first cloning of the DNA into the vector, and then subsequently the expression of the gene product. So, this is in brief the general scheme of a recombinant DNA technology. Now, what are the advantages of using recombinant DNA technology over traditional drugs? Let us focus about therapeutics only. For example, it is helpful in targeted therapies. Biopharmaceuticals are designed to target specific molecules, reducing side effect and improving treatment effectiveness. There are fewer side effects. Biopharmaceuticals have fewer side effects due to their specificity, unlike traditional drugs that affect multiple systems. And then we can also go for customized treatments. The ability to produce personalized medicines like human insulin offers tailored treatments for individual patients' needs. And then there is also economic significance. For example, in the case of revenue generation, new drugs can generate billions in revenue, and bringing drugs to market faster than can improve cash flows substantially. Then we can also go for cost reduction. For example, AI driven drug development can reduce costs in the preclinical phases by about 70% relative to traditional techniques. And then over the last 2 years, around $102 billion was invested in biotech and drug startups, and a further $7 billion was invested in tools for drug discovery. We have precision in gene manipulation when we are using this particular technology. Recombinant DNA technology allows for precise manipulation of genetic material, enabling the creation of specific gene sequences and the study of gene function. Then there is the advantage of scalability and cost effectiveness. The technology is scalable and cost effective, making it possible to produce large quantities of proteins for research and therapeutic purposes. It offers versatility in protein engineering. rDNA technology enables the production of a wide range of proteins, including human insulin, epoetin alpha, and human growth hormone, which have significantly improved treatments for various medical conditions. It helps us in achieving targeted therapies. It has led to the development of targeted therapies, reducing side effects and improving treatment effectiveness, which I have also discussed earlier. Then we are able to offer customized treatments. Now, let us discuss about the basic steps in rDNA technology. We have to go for selection of vector. It may be a cloning vector or it may be a expression vector. On the other hand, we have to also think about isolating the target gene. Then we go for cleavage of the isolated DNA at particular site by restriction enzyme. We make a cut in the vector, and we also need to digest the DNA chromosome where our gene of interest may be lying. So, this we have already briefly described in the general scheme. Then we go for the ligation of the digested target gene with the vector DNA, then introduction of gene loaded vector into compatible host cell. This is something very important. The host cell must be compatible to receive the gene loaded vector, and we have certain treatments by which we can make the host cells compatible or competent. So, that step is also known as competent cell preparation step. Then we go for replication and expression of rDNA within the host cell, and we also have to then go for screening of the transformed cells, because not all of the cells will be transformed, which means not all of the host cells, even though they are competent, may not be able to receive the gene loaded vector. So, we need to separate out the host cells which have the gene loaded vector from the host cells which do not have the gene loaded vectors. In our subsequent steps, we will be using only the cell population which have the gene loaded vector. So, we start with the isolation and purification of DNA, isolating the target gene and selecting an appropriate host for cloning is crucial. DNA from any tissue source is suitable as long as it is not degraded. Then we have the target DNA which can be obtained through classical isolation methods by established protocols, or we can also go for in silico design of our DNA sequence, and then we subsequently synthesize it in the laboratory. We get the DNA either by the methods of traditional isolation techniques or by in silico and in vitro synthesis. Then we go for polymerase chain reaction to amplify the gene of interest. Now, these figure shows the map of a vector of a commonly used cloning vector PBR322. So, some of the important features over here is that each vector has a origin. This is the point or sequence from which the replication of the vector starts. Then it has many marker genes. For example, this is ampicillin resistant gene, and this is the tetracycline resistant genes. These resistance antibiotic resistance genes helps us in screening the vector loaded host cells. So, we use these in a various steps. First, for the successful entry of this vector into the host cell, and then for the selection of a gene loaded vector inside a host. Then you can see here certain sites for restriction enzymes. You have ECO R5 here, then you have ECO R1, BAM-H1, SAL-H1, then HIND3 and PST1. So, these are the sites along with the number. So, these particular sequences will be cutting the vector in these particular sites to open it up. So, we may use BAM-H1 single enzyme to open it up. So, if we use BAM-H1, the vector will open at this site at 375. But if we can also use a combination of restriction enzymes. For example, if we use ECO R1 and SAL1, so we will have a vector which is ranging from 4359 and 651. Then we will have another fragment here, this smaller fragment. So, this fragment will be lost because it does not have the origin, but this fragment will be useful for us. So, we create a big gap over here, and in this gap we can clone actually a very big gene sequence over here. So, this is basically what a vector is all about, and the various restriction sites, the antibiotic resistant genes which helps us in the screening process, as well as the origin of replication. So, let us look into the cleavage of DNA into particular sequences. So, here the example is for BAM-H1 recognition sequence, which is basically a palindromic sequence. So, when BAM-H1 will cut any given DNA, so it will create a site like this, as you can see over here. So, once isolated, both target and vector DNA are cleaved to generate fragments of defined length or with specific endpoints, as I was showing you, from 4300 to somewhere around 400 or 300. The DNA fragment of interest is called the insert DNA, and then the DNA is cleaved by treating with the restriction enzyme, which I have already shown you. These restriction enzymes act as a molecular scissor that cut DNA at specific locations. These reactions are called restriction enzyme digestions. Then we go for the ligation of the DNA fragments. Target DNA is treated with restriction enzyme to generate fragments which are then mixed with vector DNA pre-treated with the same enzyme and DNA ligase. So, here due to complementarity of these two ends over here, they will pair with one another, and then the two molecules will be joined by DNA ligase. DNA ligase covalently joins the DNA fragments and vector DNA through 3' to 5' phosphodiester bonds. The ligation mixture contains various interconnected DNA molecules, including the vector bonded to the target DNA. Once the vector is loaded with the target DNA or insert DNA, we go for the introduction of the rDNA into a suitable host. To enable replication, the recombinant DNA molecules need to be introduced into a compatible host cell. This process of directly introducing foreign DNA into a host cell is termed genetic transformation or simply transformation. So, this is the plasmid DNA, and there is basically a loaded DNA over here. Okay. Then this plasmid will migrate towards the cell, and DNA will adsorb to the membrane here, as you can see, and then it will enter by the process of translocation across the membrane. So, many times this is difficult. So, we have to create the conditions for plasmid DNA to enter this host cell. Host cells capable of taking up the DNA are called competent cells. So, one of the facilitation that we do is treat the cells with certain chemicals which make it competent via chemical shock or even electroporation. So, when we give very high voltage or short pulse, short time, or very say low voltage for little bit extended time, because of the fluidity of the cell membrane, there will be opening of creation of pores in the membrane, and these plasmid can enter through these small pores into these competent host cells. Alternatively, recombinant DNA can be delivered to host cells via transfection, where it is encapsulated in virus particles. So, let us now study a little bit about the expression of rDNA in the host. So, various organisms can act as host for gene expression requiring specific elements in the expression vector. So, this is a vector map of a commonly used expression vector PISX3X. So, here we basically have a promoter which is very essential for a expression vector. Then we have multiple cloning site over here, and then this is a marker gene for beta lactamase, and this is a Lec repressor. So, these are the some of the basic features of an expression vector as shown here. We can classify the expression systems as prokaryotic expression systems, or eukaryotic expression systems. Prokaryotic expression systems are favored over eukaryotic ones for a fast breeding, low cost, high expression levels and easy purification of products. Eukaryotic systems has the advantage that human therapeutic proteins may show better expression, and the proteins show better folding due to presence of post-transcriptional modification machineries which are absent in the prokaryotic expression systems. So, let us look into the application of E. coli as an effective expression system. Here you can see the image of E. coli taken under a scanning electron microscope. Escherichia coli is widely used for protein production due to its fast growth, low cost, high expression levels and ease of product purification. Extensive research on E. coli genetics makes gene manipulation for recombinant protein expression simple and straightforward. However, some proteins may face challenges including difficulty in expression, the need for specific post-transcriptional modification, for example glycosylation, and the formation of inclusion bodies due to improper folding. So, we have certain engineering strategies in E. coli for overcoming glycosylation. While human cells are used to produce glycosylated proteins, E. coli can still produce non-glycosylated proteins when glycosylation is not required. For example, we have the recombinant human interleukin 2 or IL-2 expressed in E. coli which retains biological activity, but has solubility issues leading to aggregation and also immunogenicity. These issues has been addressed by mutating asparagine residues in the N-glycosylation sites to lysine, increasing the protein's isoelectric point and thereby reducing aggregation. Alternatively, mutating these sites to cysteine reduces aggregation and improves in vitro glycosylation, as in the production of recombinant human erythropoietin in Escherichia coli. Let us now discuss about other engineering strategies for overcoming other kind of post-translational modifications. Let us begin with the proteolytic cleavage. Eukaryotic proteins such as insulin are often produced as precursors, which we call as proproteins, that require proteolytic modification for activation. Insulin gene encodes a signal peptide beta chain, B chain, C peptide and A chain. Pre-proinsulin undergoes cleavage to remove the signal peptide and C peptide, leaving the A and B chains connected by two disulfide bridges. In recombinant human insulin production, the A and B chains are expressed separately in E. coli, followed by in vitro disulfide formation, which we will discuss in a later slide. Then another important PTM is disulfide bonds formation, which possess challenges in E. coli due to its reductive cytoplasm. However, proteins can be directed to the periplasm, where an oxidative environment facilitates disulfide formation, bridge formation. So, we will discuss this, see this in the slide number 35. So, protein engineering for improving stability is one of the other engineering strategy. Protein engineering is frequently used to enhance the stability of recombinant human proteins produced in bacterial systems, often by modifying cysteine residues to influence disulfide bond formation and thereby reduce aggregation. For example, cis-125 serine mutation in L-leukin, recombinant interleukin 2, and cis-17 to serine mutation in beta-ferron and beta-seron, which are recombinant IFN beta-1b products, stabilizes the protein without affecting its biological activity. Another approach to stabilize proteins involves removing unstable hydrophobic stretches exposed to water, which can cause aggregation. For example, in polypharmin, which is recombinant keratinocyte growth factor used to prevent mucositis during cancer treatments, deleting 23 N-terminal residues significantly improves stability without compromising biological activity. The next important step is the identification of host cells that contain rDNA. After the vector is loaded with the gene of interest, and then the recombinant DNA is introduced into the host cell by the method of transformation or transfection, it is essential to identify the host cells that contain this recombinant DNA molecule. So, this recombinant DNA introduction into the host organism is a low efficiency process with only a small fraction of cells successfully taking up the DNA. So, we have to separate out these transformed cells, which we will multiply and increase the population. So, for doing this, we need to go for a selection or screening process. Now, here in these genetic selection, cells are grown under conditions where only those containing the recombinant DNA with a selectable marker gene can survive and proliferate. For example, here we are using ampicillin resistance. So, these media will have ampicillin resistance, and the cells which have these vector will be able to survive in these media. So, the bacterial host antibiotic genes are often used as the selectable markers, while in yeasts selectable markers make the transformed cells auxotropic, allowing them to grow on millimole media. Let us now go for discussing the application of rDNA technology in pharmaceuticals. In this section, we have an overall overview of the application of rDNA technology in pharmaceuticals. Then we discuss about human protein replacement strategy like insulin, human growth hormone, recombinant antibodies, interferons, enzyme replacement therapy, then other recombinant enzymes and recombinant therapeutic proteins. And we will also have a small discussion on the ethical considerations, because this is all about human therapy. So, in this picture we can see novolin is a kind of an insulin, and then we have Twinrix, a vaccine comprising of inactivated hepatitis A vaccine and recombinant hepatitis B vaccine. So, recombinant DNA technology has revolutionized medicine by enabling the production of over 400 recombinant proteins, with around 50 approved for human use already. This technology is also used to identify new genes, understand their roles in diseases and in gene therapy, to treat genetic disorders, cancer and other diseases. In India, 9 products have been approved for marketing, like insulin, alpha interferon, which is a cancer drug, hepatitis B vaccine, GM-CSF, G-CSF, blood clotting factor 7, erythropoietin, this drug used in kidney failure, streptokinase, drug administered in heart attacks, and human growth hormone. Some of the key milestones in recombinant drug development, starting from 1982, when recombinant human insulin was made, which is the first recombinant biologic to be approved. And insulin, we know, is a therapy for diabetes. Then 4 years later, Muromonab CD3, which is the first approved monoclonal antibody or Murim antibody, was used for the prevention of organ reduction. Then in 89, epoetin was used for the treatment of anemia. This is the first erythropoietin to be approved. Then we have filgrastim in 1991, it is the first colony stimulating factor which was approved, used for chemotherapy induced neutropenia. Then we have abiximab, which is the first approved chimeric monoclonal antibody used against glycoprotein 2B 3A antiplatelet therapy. Then we have the daclizumab in 1997, which is the first approved humanized monoclonal antibody. So, this is a murine antibody and this is a humanized antibody. So, a lot of the sequences similar to human genetic sequences have been replaced over here. This is basically anti-rejection drug. Then we have infliximab, this is the first approved tumor necrosis factor inhibitor, and it is a chimeric antibody. This is a therapy for biologic disease modifying anti-rheumatic drug in 1998. Then we have in 2001 adalimumab, which is the first approved fully human monoclonal antibody. So, this is a humanized monoclonal antibody, but this one is a fully human monoclonal antibody. So, we have then ipilimumab, which is the first approved monoclonal antibody immunotherapy. Then we have pembrolizumab, which is a treatment of melanoma skin cancer, and it is the first approved monoclonal antibody programmed cell death 1 PD-1 inhibitor. Then we have elirocumab, which is a treatment for hypercholesterolemia. This is the first approved monoclonal antibody against proprotein convertase subtilisin caxin type 9. Then in 2016 we have atezolizumab, which is a therapy against cancer, and this is the first approved monoclonal antibody program death ligand inhibitor. So, this table has been adapted from a new type of blockbuster, The Growth of Biologics, published in 2018. So, we can see since 1982 to 2016 and even now later on, a lot of antibodies which are all recombinant, produced by the recombinant DNA technology, has been coming to the market regularly. These actually showcases the importance of this technology, rDNA technology, in pharmaceuticals. Now, let us discuss about human protein replacement strategy. Genes govern the synthesis of proteins within the cell, and any defect in the genes can lead to the production of misfolded proteins, or sometimes even the inability to produce any protein at all. In other cases, such genetic abnormalities may cause the body to not be able to produce sufficient amount of a given protein. Such gene defects are linked with inherited or genetic diseases. rDNA techniques allow us not only to identify these proteins, but also to manufacture these proteins. The manufactured proteins can then be used for the treatment of the disease, and you can already see many of those in the earlier slide. The introduction of recombinant proteins to tackle this deficiency in the human body is referred to as human protein replacement strategy. The hormone insulin was first developed using rDNA technology and has been crucial medication in treatment of high blood sugar. In medical terms, insulin refers to any pharmaceutical preparation containing the hormone insulin, which is used to treat high blood glucose conditions such as type 1 diabetes, type 2 diabetes, gestational diabetes, complications of diabetes, and hyperkalemia, which is high blood potassium levels. So, this is Humulin R and this is Actrapid. These are two synthetic insulin drugs which are available in the market. Traditionally, before these synthetic drugs, insulin was derived from pigs and cows and were used for treatment of high blood sugar levels, but this has now been replaced with human insulin production using the rDNA techniques. In 1983, Humulin, which is a brand name for recombinant human insulin produced in E. coli, developed by Genentech and licensed to Eli Lilly, became the first rDNA drug to be manufactured. We have various types of insulin. Variations of human insulin molecule called insulin analogs are also now available as alternate medications. Those bind onto insulin receptors, bringing about the same reactions as insulin. Four main types of insulin are widely available as medications. Number one is the fast acting or rapid acting, that includes insulin analogs such as Aspart, Lispro, and glulicin. They take effect within 5 to 15 minutes and remain active for 3 to 4 hours. Then we have short acting, which is basically regular insulin, which starts working in about 30 minutes and lasts for 5 to 8 hours. Then we have intermediate acting, which includes NPH insulin, which becomes active within 1 to 3 hours and has a duration of 16 to 24 hours. Then we have long acting insulin that includes analogs like Glargine U100 and Detimir, which begin working in 1 to 2 hours and provide steady activity for around 24 hours, though these can vary between individuals. So, this is a PyMol or computer generated diagram of an insulin Lispro duodecamer, which is basically a model and not a real picture of a protein. So, how insulin is produced using rDNA technology? Diabetes is a very common disease nowadays across the globe, and in India the cases of diabetes are increasing every day. In fact, we have a large diabetic population, and India is often referred to as the diabetes capital of the world. The current approach for insulin dependent diabetes is the application of synthetically prepared insulin, basically through the process of rDNA technology. So, let us try to understand how these recombinant DNA technology is useful in producing insulin. So, basically we take a expression vector which is a promoter, and then the insulin gene is cloned into this vector, and we clone the A chain and B chain into two separate vectors. So, in these we are cloning the insulin gene A subunit, and in these we are cloning the insulin gene B subunit, and both have antibiotic resistant genes, which helps us in the selection process. Once the vector is internalized into a competent cell, the insulin A is produced as a fusion protein along with the beta galactosidase, which is also under the control of the same promoter. So, in one population we will be getting the fusion protein that contains the insulin A gene, and in other we will get the insulin B fusion protein. So, these are purified separately, and the beta galactosidase is removed by a chemical method to release the A chain and the B chain. These A chain and B chain are then cross-linked with one another by disulfide bond to form a functional insulin molecule. So, these is a very popular method now, and also economic method to produce insulin in bulk qualities. So, basically you can see the recombinant plasmid separately transformed into E. coli host cells containing the A and B side chain genes, and the pro-insulin chains, that is fused beta galactosidase A chain and B beta galactosidase B chain are separately produced in the host. Then these are separated from a beta galactoside by treatment with cyanogen bromide. A and B chains are joined in vitro to produce the naive insulin by sulfonating the peptide chain with sodium disulfonate and sodium sulfite. Let us now discuss about the human growth hormone. Human growth hormone is a hormone comprising of 191 amino acids secreted by the somatotropic cells of the pituitary gland. These hormones stimulates growth, cell reproduction and regeneration in humans, and deficiency can lead to developmental disorders in children. Recombinant human growth hormone is often a treatment for growth deficiencies, Turner syndrome and chronic renal insufficiency. So, here you can see recombinant human growth hormone sold under the brand name cryptoprin. This is prescribed for children with growth hormone deficiencies. So, here is the process for the production of human growth hormone using rDNA technology. E. coli lacks the biochemical machinery to remove hydrophobic leader peptides, which are some 20 amino acids from the pre-hormone. So, you have these human growth hormone gene, and we undergo restriction digestion, and these are the synthetic oligonucleotides. So, we use T4 DNA ligase to ligate these 24 nucleotide residue with this portion of the human growth hormone, and then these are put into a vector, as in the case of insulin. So, this vector has a promoter and a multiple cloning site, and then we use restriction sites in this MCS to open up this vector, so that these construct can be loaded. So, these are then transformed into the cells and then expression host, and the AGS is produced, and then we purify the human growth hormone from the lysates. The rest of the production procedure follows similar scheme to any other recombinant product, as in the case of insulin. So, 3 nucleotide sequences chemically synthesized for the first 24 amino acids, ligation of the fragments by T4 ligase to generate the whole sequence of 84 base pairs. Then we go for the isolation of mRNA for human growth hormone. cDNA is obtained through reverse transcription, which is treated with EcoR1 and HER3 to remove the sequence for the first 24 amino acids. Ligation of the 2 DNA sequences by T4 ligase and insertion to the plasmid vector, and then recombinant plasmid is transformed into E. coli host, where the recombinant human growth hormone is produced. Now, let us discuss about the production of recombinant antibodies. Recombinant antibodies are synthetic fragments of antibodies produced by using recombinant antibody coding genes. While they mainly serve as a tool that targets antigens with high specificity and low immunogenic response, maintaining these properties over time, recombinant antibodies can be modified to incorporate effector functions such as triggering immune responses or blocking signaling pathways. In cancer treatments, recombinant antibodies can be used as a part of target modules, which can direct therapeutic agents to specific cancer cells by targeting a given antigen. Recombinant antibodies can also neutralize HIV, SSV viruses by binding onto the surface receptors, thereby compromising their ability to enter into the host cell. Recombinant antibodies can also be used as diagnostic tools, as these antigens can accurately bind onto certain target antigens. So, what are the types of recombinant antibodies? So, this is a canonical figure of full length antibody, roughly around 160 kilodalton. So, you have the variable heavy chain and variable light chain, and constant heavy chain and constant light chain over there, those portions. So, we have single chain variable fragments. Single chain SCFB is the smallest of the recombinant antibodies, molecular weight around 28 kilodalton. So, here if you consider this particular portion and then you link them covalently through a linker, you have this single chain variable fragment. They consist of the light chain VL and the heavy chain VH here, as I have shown you, which is the variable region of an immunoglobulin linked by a flexible peptide linker. Then you have the fragment antigen binding region, Fab region. Structurally, Fab fragments consist of one set each of variable and constant fragments components, which create 2 polypeptide chains. Fab fragments are anti-idiotypic antibodies, meaning they can bind directly to the paratrope of the target antibodies. So, when we are taking this entire thing, this is the Fab, which is roughly around 55 kilodalton. Then we have diabodies or bispecific recombinant antibodies combine 2 different antigen binding regions within the same molecule. The bispecific antibodies can cross-link the target molecules to 2 different cells and can directly mediate cytotoxicity. So, how these antibodies are produced using rDNA technique in E. coli. So, let us have a small discussion on that. Bacterial expression systems are extensively used for producing recombinant antibodies due to their rapid growth on low cost substrates, capacity for achieving high cell densities, well characterized genetics and ease of genetic modification. The production generally follows a similar scheme to any other recombinant product and begins with determining the sequence of the variable heavy and variable light genes, followed by cloning these variable regions into a plasmid vector. So, we have this variable heavy and variable light, and these are introduced into plasmid, and these are transformed into E. coli. Then these are expressed, and then you have these followed by extraction and purification of the antibodies. So, after transformation, we grow them under favorable growth conditions. One of the major issues pertaining to production in E. coli is the lack of protein folding mechanisms, which may lead to formation of inclusion bodies. To address these, two strategies have been adopted: periplasmic secretion and in vitro refolding. So, here this is the plasmid containing the gene. So, after transcription it is translated and the protein is produced. So, here this particular protein is allowed to go for periplasmic secretion, and or it is here it gives the functional recombinant antibody. Then in the other case it goes into the cytoplasmic inclusion body, which we can also separate out. So, once it is in the periplasm, we go for the separation, isolation, and then we allow for in vitro refolding. A second approach to obtain this functionality involves mimicking eukaryotic antibiotic secretion in E. coli, where proteins with a signal sequence are directed to the periplasm. Periplasmic space having an oxidizing environment opposed to the reductive cytoplasm oxidizes the sulfahydryl groups, enabling the formation of the disulfide bonds over here, as you can see. However, high level expression may lead to insoluble aggregates due to folding intermediates. Protein aggregations can be resolved in vivo by co-expression of periplasmic chaperones like SKP, OMPH. Aggregations may also be resolved in vitro using the methods as follows. The first approach to production of recombinant antibodies in E. coli is to produce antibody protein as cytoplasmic included bodies, followed by in vitro refolding. In this case, the protein is expressed without a signal sequence under a strong promoter. The inclusion bodies contain the recombinant protein in a non-native and non-active conformation, which is then refolded using any of the following methods. We may have dilution refolding, we may have redox refolding or disulfide restricted refolding. In the case of dilution refolding, we have around 12.6% efficiency, redox refolding 40%, disulfide restricted refolding 50%. So, from the point of view of reinitiation type, dilution refolding, here reinitiation prior to disulfide bond formation takes place, and the various steps involved here are deinitiated proteins are first solubilized in a high deinitiatant concentration, then the deinitiatant concentration is gradually decreased to dialysis. Then we have the redox unfolding, where reinitiation and disulfide bond formation happens simultaneously. This utilizes a glutathione redox coupling system to catalyze disulfide interactions as the protein refolds into its native state. Then we have these disulfide restricted refolding, where disulfide bond formation prior to reinitiation takes place, where deinitiated proteins are first solubilized agents that catalyzed disulfide bond formation, after which it is refolded by the slow removal of the denaturing agent. Now, let us look into some of the FDA approved recombinant antibodies produced in E. coli. We have the generic name in the first column, followed by the molecular format, then expression condition, and the disease against which this was developed and the year of approval. So, we have sartolizumab pegol. This is basically pegylated FAB prime, soluble in the periplasm. It is used against the Crohn disease, developed in 2008. Then we have ranibizumab. This is basically Fab soluble in periplasm. This is against the wet AMD developed earlier than the pegylated Fab in 2006. Then there are others which you can simply go through this table, and the latest ones are the Tebentafusp or Kimtrac. This is a bispecific TCR SCFB fusion. This is a expression condition is inclusion body refolding in the cytoplasm used against metastatic uveal melanoma. Let us discuss about the production of antibodies by rDNA technique, the fast display method. So, fast display is a widely used technology for recombinant antibody production, where the target antibody is expressed on the surface of a bacteriophage. The process begins by determining the sequence of the VH and VL genes, which are then cloned into the genome of the M13 filamentous phage. The modified phage genome is introduced into E. coli via transduction along with co-infection by helper phages. This co-infection enables the replication, packaging and assembly of virions, each displaying diverse antibodies on their surfaces, forming a phage library. So, here are the extracted VH, VL genes. So, we clone them, and then there is a co-infection with helper phage DNA over here, and then there is a production of phage with displayed antibodies, and then we get a phage library where you see the various antibodies are getting displayed. From this library, desired monoclonal antibody can be selected by target immobilization using microtiter plate well. So, here you have the phage library, then we go for the target immobilization, then washing of the bound phage over here, and then the unbound phage will be washed off, while those which have been captured by this plate will be retained, and then these are eluted out, and these are then further amplified, or we can go for a repeat of this panning process for around biopanning 3 to 5 cycles. The selected antibodies can then be used to produce more phage by co-infecting bacterial cells with helper phages, and through repeated cycling of these steps or panning, desired antibodies are obtained. Phage obtained in the final step of biopanning is used to infect a suitable bacterial host, from which the antibodies can be purified and analyzed. So, here is a list of recombinant antibodies produced using the phage display technology. So, in the first column you can see the name, for example, adalimumab, and then in the second column you have the company or the developer, this is FY, and then you get the target, this is TNF alpha, and the indication against which it is useful, like rheumatoid arthritis, Crohn's disease, psoriatic arthritis, plaque psoriasis, and so on, and status or the trade name, this is approved that as Humira. So, this table you can go through, and you can see many such antibodies has been developed, whether it is against systemic lupus or squamous lung cancer, then asthma, then Alzheimer's disease, and also as a prophylaxis and treatment of anthrax, and some of them are already approved, while some of them were in phase 3 trials when this table was compiled. Let us now go for discussing about the interference, which are naturally occurring signaling proteins secreted by eukaryotic cells in response to viral infections, which when bound to receptors on adjacent cells trigger the production of proteins that inhibit viral protein synthesis. While primarily acting to suppress viral infections, IFNs also stimulate immune cells and enhance host defense by elevating antigen presentation. Interference may thus be used in control of a wide range of diseases, including viral infections, autoimmune diseases like multiple sclerosis, and can also be used in combinatorial therapy with radiation and chemotherapy for treatment of cancer. So, here we discuss about the production of interference using the rDNA technique. The synthesis of IFNs using rDNA technique was successful with the insertion of the sequence coding for human leukocyte IFN or IFN alpha to the yeast alcohol dehydrogenase gene in a plasmid. The recombinant DNA was introduced into cells of Saccharomyces cerevisiae. In the earlier case, we were mostly using E. coli, but here we are using yeast, and the host cells could synthesize large amounts of interferon. Synthesis is possible in E. coli, but production is relatively low and took more time as prokaryotes lack the machinery required for post-transcriptional modifications that are required for human proteins. Some of the recombinant interferons available for therapeutic uses are listed in this table here. So, you have the brand name and the type in the second column and the use. So, the brand name is Alpheron N. So, here this is human leukocyte derived interferon alpha entry, and it is used against genital and perineal warts. Then you have Roferon A, which is recombinant interferon alpha 2A. Then it is used against hairy cell leukemia and also in acquired immunodeficiency syndrome. Then you have Intron A, which is recombinant interferon alpha 2B against hairy cell leukemia and AIDS. Then Evonex, Rebif, which is recombinant interferon alpha 1A, used against multiple sclerosis, and against multiple sclerosis there is another drug which is the beta-seron. Basically it is a recombinant interferon beta 1B. So, next we go to discuss about enzyme replacement therapy, ERT, which is a medical treatment devised in the 1960s by Christian D. Duve and Roscoe Brady. This involves the intravenous administration of an enzyme for which the patient is deficient or it is absent in the body of the patient. ERT does not treat the genetic disorder causing the enzyme deficiency. It increases the concentration of the enzyme that is lacking in the patient. ERT treatments are now available for some lysosomal storage diseases such as Gaucher disease, Fabry disease, MPS 1, MPS 2, Hunter syndrome, MPS 4 and Pompe disease. So, here in this picture you can see some drugs used in enzyme replacement therapy. In the top you can see Fabrazyme, a medication for Fabry's disease. Then you have this Cerezyme over here, a medication for Gaucher disease. ERT has also been used to treat patients with severe combined immunodeficiency resulting from an adenosine deaminase deficiency. So, we have numerous recombinant enzymes produced in microbes against various diseases, and the expression system ranges from E. coli, Pichia pastoris, Saccharomyces cerevisiae, then Ogachia minuta, then Yersinia lipolytica, and then we have also Saccharomyces pombe. And then we have various different enzyme targets. For example, we have adenosine deaminase against ADA-SCID. Then in Gaucher's disease we have beta glucocerebrosidase. In Fabry's disease we have alpha galactosidase. In Pompe's disease we have alpha glucosidase, and so on. Apart from these, there are many other recombinant therapeutic enzymes like recombinant uricase or urate oxidase, which is an enzyme that catalyzes the conversion of uric acid to 5-hydroxysorate and can therefore be used in treating conditions related to high amounts of uric acid in gout and in rhabdomyolysis with kidney failure. In humans, however, the enzyme is absent, and therefore recombinant uricases are widely used in treatment of hyperuricemia. Then we have rasburicase. This is produced in Saccharomyces cerevisiae, and pegloticase produced in E. coli are two variations available for commercial use. And we have recombinant carboxypeptidase G2, glucarpidase or Voraxizase is a medication used for the treatment of elevated levels of methotrexate during treatment of cancer patients who have impaired kidney function. Glucarpidase, a recombinant form of the bacterial enzyme carboxypeptidase G2, converts methotrexate into less toxic glutamate and 2,4-diamino and methyl teichoic acid. So, in this table we can see many therapeutic agents. The first column we have the drug name and then the cell factory or the organism in which it is produced. Mostly these are all majorly produced in E. coli, and then we have also Saccharomyces cerevisiae, and then this column tells you about the mechanism of action. For example, this filgrastim stimulates hematopoiesis, and it is used in the bone marrow transplantation and cancer chemotherapy induced neutropenia. Then we have pegfilgrastim. This is also called as NuPeg. This stimulates differentiation, proliferation and activation of the neutrophilic granulocytes used in cancer chemotherapy induced neutropenia. Then we have denileukin diphthy tox, which is a fusion protein. This is diphtheria toxin used fused to cytokine. Indications is cutaneous T cell lymphoma. So, kindly go across this table which have several other drugs like endostatin, denileukin diphthy tox we have already discussed, then palifarmin and so on. Now, let us discuss about one very important issue which is ethics, ethical considerations, particularly human gene therapy. Focuses on the implications of altering genetic material, particularly regarding safety, efficacy and long term consequences. Key concerns include ensuring informed consent, minimizing risk, addressing unintended genetic changes and evaluating the societal impact of such modifications. And GMO labeling and the public perception is also an important ethical issue. The labeling of genetically modified organisms raises ethical issues related to consumer rights, trust in recombinant DNA products and transparency. Some people may not prefer GMO products, and they should be given this opportunity to identify the product. Clear and accurate labeling ensures individuals are informed about the content of the medications and products, and they can therefore take an informed decision. Other important issues are regulatory oversight and responsible innovation. Regulatory frameworks are essential for ensuring safety, scientific integrity and ethical standards in gene therapy and GMO technologies. Oversight must balance innovation with patient safety, societal impacts and accountability. Responsible innovation requires transparency, stakeholder engagement and the considerations of broader societal implications. So, with these we come to end of this lecture. Thank you for your kind attention.